right, hi everyone. Thanks so much. It's a real honor to be here and speak to everyone about my research. I've been at UC Santa Cruz for 6 years now as a faculty member. So I'd like to start off with a question that I'm sure many in this room get from people, and it's why do we have a vaccine for every virus already? Why doesn't these traditional methods of either growing up the virus in the lab and inactivating it or attenuating the virus, why doesn't that work for everything? And as we all can appreciate, every virus is different and special in its own way. But some of the main reasons are that the immune responses from these traditional vaccines are just either they don't elicit protective immunity, or they might have safety issues, either side effects or in really bad cases, vaccine-enhanced disease. Another reason might be that it's just hard because we either can't grow the virus in the lab, or not very well, or we don't have appropriate animal models. And so I'd like to make the argument that rationally designed vaccines, such as subunit vaccines, which is really just recombinant virus proteins made in the lab, can overcome a lot of these challenges. And I'm going to tell you two stories about rationally designed subunit vaccines in my lab. So here are the questions my lab asks. What do virus antigens look like at the molecular level? We mainly use X-ray crystallography, but are dabbling in cryo-EM and NMR as well. Where do protective or neutralizing antibodies bind to these antigens? What are the important epitopes on the surfaces of viruses? And how do these antibodies work to protect from infection and disease? And can we use this molecular information to design subunit vaccines that elicit protective immunity? So the first project I'll talk about is human astrovirus, led by two wonderful graduate students, a former grad student, Walter, and Lena Meyer, who has a poster today. So human astrovirus is a leading cause of viral diarrhea and vomiting in children. And so every one of us in the room has been infected, usually as children, millions of infections worldwide. And I don't think it was really appreciated until recently that the disease severity of astrovirus infection is really on the same level as norovirus. There are 8 canonical serotypes, 1 through 8. I'll mention that the host cell receptor or receptors is unknown for this virus, and there are no vaccines or antiviral therapies. But I think there's high potential to develop a vaccine. First, the rarity of astrovirus infection in healthy adults. Again, most of us are seropositive. In one clinical study where adult volunteers ingested astrovirus, those that had low or no antibodies had more severe disease. And finally, there's evidence for immunoglobulin therapy being protective. So when I started this project, this is what was known. It's a really simple single-stranded RNA virus with one capsid protein that forms the shell. And what was known is that host proteases cleave the capsid both intracellularly and extracellularly, and you get this release of some of the spikes on the surface of the capsid, and this forms the infectious particle. And if you run this on a gel, you get 2 bands. And so looking at this capsid protein, we wanted to know what it looks like. And instead of making recombinant versions of these 2 proteins, we took a structural approach and asked what are the structural domains. And so using HHPRED, it's an online sort of structure prediction program, we designed some constructs that we predicted would make sort of the core of the capsid and the spike. We expressed these in E. coli, and we're able to purify soluble versions of these and determine their structures by X-ray crystallography. And so the core domain, which forms sort of the icosahedral shell of the virus, is very similar in a lot of different viruses. It has most structural homology to hepatitis E virus. And then the spike domain over here forms a dimer on the surface. And so we modeled these structures into the cryo-EM maps of the virion particle, and we really learned a lot about virus maturation, this protease maturation, and evolution. But what I'd like to focus on is the question of where do these neutralizing antibodies bind? This is what our immune system sees when we're infected. And so we dug through the literature, and we found an old paper that had isolated a monoclonal antibody this antibody neutralizes virus infection in cell culture. And I contacted the corresponding author, and she said, well, we lost the hybridoma cells in freezer damage, but we still have some of the antibody left over. And then I said, sure, I'd like to study it. So we purified the antibody. We used ELISA to show. What we found is that the antibody targets the spike on the surface of the virus. And it's specific for serotype 2, which we're calling spike 2, and does not bind to the other serotypes, and it also does not bind the core domain. We also determined affinity, low nanomolar binding affinity. Little side story, we didn't have enough of the antibody to do further structural studies, so we had to do some de novo sequencing. But with that sequence, we were able to generate synthetic genes for the antibody, and a lot of times what we use in my lab are single chain variable fragments, just mini versions of the antibody. These are very amenable and useful for crystallography. We can make them pretty well in stably transfected insect cells. And in this case, the SCFV, the recombinant SCFV, still had the same specificity and binding affinity as the original antibody. So we solved, this was our first antibody antigen structure complex that we solved in the lab. And this is the first time an epitope has been defined on the surface of the virus. And what I'd really like you just to appreciate is that, so we have two antibody SCFVs binding to the spike dimer, and the antibody binds to a large surface, and it's a three-dimensional epitope. It's binding not just a linear sequence, but multiple loops coming from different parts of the spike protein sequence. We have since solved another structure of spike antibody complex in the lab. That's Lena, who has a poster. And what you can see is that this antibody binds in a completely different site. Again, both of these are neutralizing antibodies, and it's pretty cool to see that, to guess, are these blocking potentially the same functional site that's in the middle of the two epitopes, or are these blocking two different functional sites on the surface of the spike? And so to test the functionality and mechanism of these antibodies, we're developing some assays. And we wanted to ask the question, is the spike a receptor binding domain? The spikes are often receptor binding domains in other viruses. And so the first experiment we did in collaboration with the Arias lab at the University of Mexico is we incubated CACO2 cells. They're a colon cancer cell line that is susceptible to astrovirus infection. We added spike to the media and asked, does it block virus infection? And sure enough, increasing amounts of spike were able to block virus infectivity in cell culture. And interestingly, spike from one serotype was able to block infectivity by different ones, suggesting possibly that they might use the same receptor. In my lab, we also generated GFP fused to spike and asked, does it bind to the cells? GFP spike is still recognized by the antibody. And then we use flow cytometry. And so here is what it looks like just with GFP alone. We get this curve of the cells. And any of the cells that were incubated with a spike fused to GFP have this long trail of fluorescent cells. And when we incubate the cells with a CFV that's specific for that spike, we get back to background levels. So this suggests that the spike alone is able to attach to cells. We have a little bit of microscopy data showing that spike GFP is binding to cells and potentially even getting inside cells. So it might be a mechanism not only for viral attachment, but viral endocytosis. So getting back to the vaccine stuff, we asked, since spike is recognized by neutralizing antibodies, will recombinant spike be able to elicit neutralizing antibodies? And we tested both recombinant spike and core. And then taking the sera from immunized mice, we asked, does that sera neutralize astrovirus infection? And this is just 2 mice, but so far what we find is that mice immunized with spike generate neutralizing antibodies, where mice immunized with the core do not. They do generate antibodies against the core, but those antibodies are not neutralizing. So we think we have the basis for an astrovirus vaccine. And so future studies in my lab are, of course, solving more antibody spike structures and defining the epitopes, determining the mechanisms of antibody neutralization. We're on fishing expeditions to identify the receptors. We're also interested in these divergent strains of astrovirus that have been found in a lot of fatal cases, or not a lot, a handful of fatal cases of encephalitis. And these are divergent at the spike domain sequence. And then, of course, our interest in developing a subunit vaccine. So switching over to our second project, this is headed by two wonderful graduate students, a research specialist, and an undergrad who's also here today. So this is a project that's also in collaboration with Trellis Bioscience, who isolated some of the monoclonal antibodies I'll talk about. So many of you know respiratory syncytial virus. It's a top cause of severe lower respiratory tract infections in children. Causes thousands of child deaths each year worldwide, and this is just really devastating. What we have right now is a anti-F protein neutralizing antibody called palivizumab. It's given prophylactically to high risk infants. And while this does not fully prevent RSV infection, it decreases disease severity. So we have no post-infection treatments, antiviral treatments, and no vaccines for this virus. So this is an envelope, a lipid enveloped virus with the F and G glycoproteins on the surface. The G is involved in virus attachment to cells, and F is involved in fusion of the virus and host cell membranes. So there's been a really devastating history of vaccine development for RSV. In the 1960s, there was a clinical trial with formalin inactivated RSV, and this was associated with enhanced disease in the vaccinated children that were subsequently naturally infected with the virus. And live attenuated vaccines have just been challenging to achieve this balance of immunogenicity and safety. So a big chunk of the field has been focusing on the F glycoprotein for vaccine efforts. If you take any one of us that have been exposed to RSV, if you look at our sera, the majority of RSV neutralizing antibodies targets the F protein. If you delete the G gene, the virus is still infectious. The G gene is variable in sequence, and it's heavily N-glycosylated. So the F protein starts off as a pre-fusion form, and following viral fusion, it converts to the post-fusion form. So here's the state of RSV vaccines and therapeutics at the moment. We've had a lot of failures, unfortunately, in vaccines and antibody therapies. The Novavax post-fusion F vaccine has not met endpoint goals in clinical trials. And other vaccines have failed, and anti-F antibodies have been failing. And so there's been just a ton of investment in anti-F strategies for RSV, and so far they haven't panned out. And I think a key piece of insight has come from this paper that has looked at babies who get passive transfer of antibodies from their mothers and asked, of those babies, who gets severe RSV infections and who gets mild ones, and what kind of antibodies did they get from their mother? And to a lot of people's surprise, antibodies to the post-fusion F did not correlate with protection from severe disease, although antibodies to the pre-fusion F protein did correlate, as well as antibodies to the G protein, the G glycoprotein. And as I mentioned, F protein antibodies are the most concentrated in our sera. So even at this dismal concentration of anti-G antibodies, that correlated with protection from RSV disease. So I think the old school way of thinking and focusing on F is still a valid case of, pre-fusion F is a strategy that a lot of people are still focusing on, and I think it has promise. But I do think the G glycoprotein has been overlooked as an important target for RSV treatments and preventative therapies. So remember what I told you, all the reasons why we're not focusing on the G. If you delete it, it's still infectious. This virus is still infectious in cell culture models, but it's actually highly attenuated in animal models. So cell culture models do not always mimic what happens in vivo. This G sequence is highly variable and it's heavily glycosylated, but RSV-G has a central conserved region. It's highly conserved, is the target of antibodies, and it's not glycosylated. RSV-G has an important role in sabotage of host immunity, so I think it's also an important target when thinking about disease. And as I mentioned, antibody levels against the G protein correlate with protection from severe disease. Okay, so I'd like to thank my grant reviewer for calling the G antigen the ugly duckling antigen. So this is what a model of the G glycoprotein, and all of this is glycosylation. All these predicted NNO glycosylation sites up here. It should be about 30 kilodaltons, but runs a little bit closer to 90 kilodaltons on the gel because of the glycosylation. As I mentioned, G has two key functions. It's a virus attachment protein, and it binds to the chemokine receptor CX3CR1 on the surface of airway epithelial cells and promotes virus entry. But what I think a lot of people don't know is that it's also a pathogenic protein. So about 6 hours after virus infection, cells start secreting a soluble version of the G. It has an alternate methionine over here, and this soluble G glycoprotein goes out and modulates host immunity. It modulates signaling and trafficking of CX3CR1 positive immune cells. We get the cytokine imbalance and promotes airway congestion. So I think G is really important for virus infection and virus disease. There's plenty of animal model evidence for antibodies against the G glycoprotein being protective in animal studies. You can administer either prophylactically or even post-infection treatments shows that anti-G antibodies are antiviral and decrease viral infectivity. But these antibodies also decrease disease and pathogenicity. They reduce cell influx, airway detention, they reduce mucus production, and they restore antiviral interferon alpha production that is suppressed by the G. So in collaboration with Trellis Bioscience, who is developing a therapeutic antibody that targets the G glycoprotein, I contacted them and said, hey, can I study your antibodies? And luckily they said yes. They've isolated human monoclonal antibodies that target the G, specifically ones that target G from the different clades of RSV. And they had done linear epitope mapping of just overlapping sequences, peptides from G, and found the general region where a lot of these antibodies bind. Their top one binds with very low picomolar affinity, so super high affinity. And so what I wanted to ask is, what does this region of the protein look like at the molecular level, and how do these antibodies target this, and are they protective? Or how are they protective? So first we solved the structure of the antibody 3D3 bound to its linear epitope. And what we noticed immediately is this long CDR3 loop that was not binding to anything at all. So we suspected that this antibody binds to a larger epitope than the linear epitope. And sure enough, we got the structure with a larger fragment of the G. And sure enough, it is this large conformational epitope, and the G is stabilized by 2 disulfide bonds here. We have since solved 2 more structures of antibodies bound to the G glycoprotein, central conserved region, and again, in both cases, they're conformational epitopes, and this really hasn't been previously appreciated. Some groups were trying to develop vaccines based on the linear epitope sequences, and we can appreciate that this kind of peptide is not going to elicit this kind of antibody. So if we compare all the structures, what we found is that The G protein is captured in all sorts of different conformations by these antibodies. Again, this is a highly conserved region, but we can appreciate it's a really highly flexible region. And this flexibility is probably very important to elicit diverse kinds of antibodies that target it from all different angles and conformations. We also developed an assay to look at the mechanism of these antibodies, and this is through a chemotaxis assay. So what we do is put CX3, CR1 positive cells in a trans well that has these tiny pores. And we put our G chemoattractant down here, and we count the number of cells that are able to migrate towards the G antigen. So we have full-length G ectodomain as well as our tiny central conserved domain region that we had purified in the lab. And what we find is that cells chemotax towards the G ectodomain as well as this tiny little central region. So this region is, essential for cell migration. And incubation with the antibodies completely blocks cell migration. And antibodies against that receptor also block that cell migration. So what my lab is doing now is taking all of this information and trying to develop a vaccine. So what I didn't mention is that RSVG-based vaccines are associated with vaccine-enhanced disease. So this is a big reason why no one wants to touch this antigen. So if you immunize animals with just recombinant G glycoprotein, there is vaccine-enhanced respiratory disease upon RSV infection of these animals. And this is also seen with a vaccine of vaccinia virus expressing the G glycoprotein. So what I'd like to do is molecularly tease apart the good and the bad of the G glycoprotein. So what we want to do is preserve the antibody binding sites while disrupting the receptor modulation site. And I can't tell you all the details, but what we do have is a number of mutants that have completely lost chemotactic activity. We've developed a receptor binding assay with flow cytometry. And we're also using binding affinity studies to confirm that our our vaccine candidates, although they're receptor inactive, that they still are displaying the conformational epitopes of the G glycoprotein, which is really important for their ability to elicit those high affinity antibodies. So the big picture is that I hope I've convinced you that molecular insights into neutralizing antibody epitopes provide a really great basis for the rational design of vaccine antigens. In both projects, these antigens, we've produced milligrams in E. coli, and so it's a really simple and affordable way to make antigens. Vaccine antigens should definitely preserve the native 3D structures of conformational epitopes in order to elicit these high affinity antibodies. At the same time, what I hope I convinced you is that it might also be important to preserve the flexibility of antigenic loops in order to elicit diverse kinds of antibodies. And so there is a lot of effort to really stabilize a lot of antigens and make them structurally stable, but we want to keep in mind also that flexible antigenic loops will elicit more diverse antibodies that target epitopes from different angles, and that could be important to be able to target lots of different strains of viruses. So anti-G antibodies are dual functioning. They're antiviral and antipathogenic, and so I'm interested in exploring other extracellular pathogenic viral proteins that could be vaccine targets that have been overlooked in the past. And this is basically thinking about our bacterial vaccines, some of our, like our tetanus and diphtheria. These are inactivated toxins that are vaccines, so we generate antibodies against the toxins. These are not antibacterial vaccines in that we don't generate antibacterial antibodies, we generate antitoxin vaccines. So I think that could be a cool new concept in vaccine design, is targeting some of these overlooked antigens. Okay, so I have had a great team of graduate students and undergraduate students in my lab focusing on these two projects. I've gotten donations of monoclonal antibodies to study, and I am certainly, I welcome other projects. Yes, you must have noticed an enthusiastic undergrad that was involved in solving the structure of the astrovirus core. So enthusiastic that he got astrovirus tattooed on his leg. So I'm really inspiring the next generation of scientists, I think. I have great collaborators in virology for both the astrovirus and RSV projects. We collect a lot of our crystallographic data here in Berkeley at the Advanced Light Source, and appreciate the funding for this work. Thank you for listening.
Thank you for a great talk. First question. No, go ahead.
Yeah.
Yeah.
You haven't mentioned anything about cell-mediated immunity. The reason I say that is in more relay virus, as we've shown, you could have high level of neutralizing antibody and get absolutely no protection, but it's all cell-mediated immunity directed. So have you looked into cell-mediated immunity much before in RSV?
Yes, I know that cell-mediated immunity does occur for antibodies that target the G glycoprotein on the surface of infected cells. So yes, I know that that is another mechanism of virus neutralization. Yes.
Hi, push it up. Is it on? Yes. First of all, beautiful work. That's awesome. I'm really interested in this dichotomy between preserving the floppiness of an epitope versus locking it down, especially because a lot of protein design has gone into recapitulating the bound conformations of these neutralizing epitopes. What kind of biological data could inform which approach is better? Because I've heard the hypothesis that this floppiness is also a way of evading the immune system. So how can we inform how we should be designing vaccines, and what kind of data can we gather to answer that question?
Yeah, it's a really good question. People come up to me all the time and be like, you know, we have monoclonal antibodies. It's really easy for viruses to escape. You get a point mutation, and so I am focusing on the polyclonal response. Escape to one monoclonal antibody, but it might still be susceptible to another one that targets from a different angle. So I do think that maybe in vaccine design we should be looking, we now have great technologies for antibody repertoire sequencing, so to look at the diversity of antibody responses in addition to neutralizing levels, titers, and so that's definitely something I'm interested in looking at, is repertoires and diversity of antibody responses and ultimately resistance to many different strains.
Last question.
Yeah, congratulations. It's great work, very inspiring. My question is actually quite similar to the last one. First of all, what's known about the molecular basis of enhanced disease? And then the second question related is, you said that the soluble form is somehow modified in relation to the form that remains attached to the virion, if I understood correctly. And what is the difference between the two?
Yeah, so there have been animal studies of viruses infection where the soluble form is eliminated, and there's certainly there's evidence for less disease. Of course, the viral attached form, or the membrane attached form, could still stimulate receptors in neighboring cells and things. So I will say it's complicated and is a lot of immunology, but it's on both our lung epithelial cells as well as migrating cells, CD4 cells that have this receptor are all modulated in terms of their migration, which relies on this interaction with the native ligand, fractalkine, as well as it modulates their signaling.